Hello, Mr. Allman here, and in this Allmencast, I want to answer the question of why it is that you need salt. The topics here are going to be the neuron. What is the structure of the neuron? How do neurons send signals? And how do neurons communicate with each other? So what's important to know is that the neuron is the most basic unit of your nervous system. It's making up the entirety in terms of the cell types of your brain, of your spinal cord, and of your nerves. Now, to start out, when we look at the nervous system, its job is to send signals. It's the messenger. It is receiving signals, integrating signals, and interpreting, sending out signals, telling your different parts of your body to do things. It's very fast, it's very accurate, and it's able to reset quickly. So these are these short-term messages, and it's necessary in a complex, multicellular body like our own for every part of the body to be in constant communication so that the brain ultimately can control exactly what's happening with all the other parts. It keeps you safe, helps you to know what's going on in your environment, and helps you to respond accordingly to keep you alive. Now, three types of neurons that are significant. We have different sensory organs, for example, your eyes, your skin, your ears, your tongue, and those sense organs are sending signals to your central nervous system through sensory neurons. So, for example, your eye is going to send a signal through a sensory neuron to your brain about conditions of light in your environment. Within your central nervous system, which is your brain and your spinal cord, you have relay neurons, which are also called interneurons, that are communicating one neuron to another, helping to interpret, to process, to create memories, and ultimately decide what to do with that information that's coming in. Now, on the way out, you have motor neurons. Motor neurons are sending signals from your central nervous system out to your muscles, telling them to move and exactly what to do. The structure of the neuron really helps determine its function. There are a lot of points of entry here. So we have the dendrites, of which there are going to be many, so many possible connections from one neuron to other neurons that are around. So these signals can come in, but there's only one path out. So once the signal gets in, there's only one path down the axon so that that neuron can then send the signal from wherever it came from to the next connection in the chain. Some interesting things about neurons, they are highly specialized, and both their size, their structure, what they contain is definitely unique amongst cells in the body. They are the longest cells, not necessarily the biggest, but certainly the longest cells, and we see that because the length of motor neurons, they have to go from your central nervous system out to the furthest part of your body. So, from, for example, your spine down to your toes, in a giraffe up the length of their neck, for a blue whale down the length to its tail, and those can be meters in length. A single cell that's going to be in our body a meter or two long is really significant, considering that most of our cells are microscopic. So here's your general structure here. You have the cell body containing the nucleus of your neuron. You have the dendrites, which are receiving signals, and then you have the axon. The axon is going to be the longest part. That's the part that can be meters in length. There's an axon hillock where the message is going to be sent down the axon itself, and then you have these nerve terminals at the very end of the cell. You have myelin sheath, which consists of this kind of insulation that's covering each of your neurons, and you have cells that produce that myelin that are covering the axon itself. The sheath is not continuous. You notice that there's little gaps in between, and those are called the nodes of Ranvier. So those little gaps are where the axon itself is going to be exposed, and that's necessary in order to make sure, as we'll see a little bit later in the video, how it is that the action potential can be carried along the length of the cell. So moving forward, dendrites, again, purpose here, incoming signals enter in, and there's going to be a little bit of space between the dendrite and the axon of another cell, and that's called a synapse. Now, all of these neurons live in a sea of ions. So these positively and negatively charged ions, both in and outside the cell, are there at all times. So you have, within the cell, you have a concentration of potassium ions and overall negatively charged ions. So inside the cell it's negative, whereas outside the cell it's going to be more positive. You have a lot of sodium there, and in general positively charged cations. And so This is the first instance where we see salt is important, because we know sodium and chlorine, that's the primary form of salt that we eat, sodium chloride, and we need to have those ions in and around our cells in order for them, particularly for neurons, for them to be able to send signals. We say that the membrane is polarized and it has membrane potential. What that means is that we have a distribution of charge, more negative on the outside, more positive charge on the outside, and that difference provides a concentration gradient, and there's energy that's stored in that concentration gradient that can be used to help to send signals. Now if you remember back to earlier in the year, we studied diffusion, and diffusion is the movement from a higher to lower concentration, and diffusion is something that's going to happen spontaneously. So if we have a concentration of positively charged sodium ions outside, they are going to want to get into the cell if they are allowed. And so that potential for movement is something that the cell is going to be able to utilize as part of this process. The axon itself, well, both the nerve and the neuron as a whole, and certainly the axon, as we'll see, is covered with all of these protein pumps and ion channels. So the sodium-potassium pump is an important one. It's a two-way pump, so we can the job of it is to pump sodium out of the cell, 3 sodiums out, and potassium in. So 2 potassiums will enter in for every 3 sodium that's out. The reason we need a pump is in cases when they're going to move against the concentration gradient, and so that's a form of active transport, and it requires ATP. The potassium and sodium channels, these are gates, and these gated channels will open and close at times, depending on whether a signal is being sent or not. And this is going to rely on diffusion, so this is a type of facilitated diffusion that's happening here. So to go back, what we want to take a look at now is here the axon hillock and the axon itself and what's happening at this point when an action potential begins. So action potential is going to be the way that a signal is sent within a single nerve. So from the cell body of the neuron down to the edge of the axon, it's an electrical signal. And this electrical signal, you can think of it like, so think about a set of dominoes. When you set dominoes up, though you tip over the first domino in the line. And the first domino knocks over the second one, which knocks over the third one, which knocks over all the way down the line from one end to the other. The dominoes themselves don't actually move physically from the beginning to the end of the line, but it's just a wave that is traveling from the starting dominoes through to the ending dominoes, and the signal is they're tipping over. So neurons have a similar setup, but instead of dominoes we have ions, and these ion channels are going to open to allow sodium in and potassium out, starting at the beginning of the axon and traveling as a wave through down to the end of the axon. So action potentials happen super quick. You can see time in terms of milliseconds here. So a millisecond is going to be, again, thousandths of a second. It is an all or nothing signal, so this is interesting because the sending of that signal is not something that has any degree to it. There's no strong or weak signal to it. And that's a little bit confusing because if you think of your muscles, your decision to send an action potential to move, for example, your bicep. And as you're moving your arm, you know that you can either flex that muscle really strongly if you're picking up something heavy, or you can flex that muscle weakly if you're picking up a small object or just lifting up your hand by itself. But it's not that the signal is weaker, it is the fact that your brain is controlling the number of signals that are going to be sent. So whether or not all of the different parts of your muscle need to contract to give a very strong message to your muscle to contract and pick up a heavy object, or just send a few signals to a few cells within your muscle in those cases where you only need a little bit of power to be able to lift it up. All right, now into the nitty-gritty of the action potential. So how does this work? So we start out with the neuron at rest. All the gates are closed. We have lots of sodium outside the cell. We have lots of potassium inside the cell. We're going to start out with the sodium gates. Sodium gates are going to be the first thing to be able to open. So when the sodium gates open, because there's lots of sodium concentrated outside the cell, by diffusion, all that sodium is going to start to pour into the cell, into the axon itself. So this is going to be the rising phase of the action potential. So you can see number 1 at rest, then number 2, the gates begin to open, and as the gates are fully open, number 3, that's where we change. We go from having a negative charge inside the cell, now we have a positive charge inside the cell relative to the environment outside. Then those sodium gates shut, so the sodium can no longer move in or out. So there's a lot of sodium now trapped inside the cell, that's why the cell is positive. However, this signals the potassium gates to open in response. So the potassium gates open, and because there's lots of potassium inside the cell, the potassium is now going to spill out of the cell by diffusion. And as the potassium leaves, then we go from a positive environment back to a negative environment. The final step here is that the potassium gates will close, and now the pump needs to restore the original potential. Put the sodium, make sure that the sodium will now exit the cell, and the potassium will be brought into the cell. So to look at that again, number 1 is the resting potential, so negative inside the cell. We have the stimulus. The stimulus comes from the dendrite. The stimulus gives the signal for the sodium gates to open. Sodium gates open for number 2 and number 3, so now sodium is inside the cell is now positively charged. Then this is known as the depolarization phase. Then the falling potential, so the potassium gates open, sodium gates are closed, potassium exits the cell becomes negatively charged again as all that potassium leaves. Now we have to switch, and we need to reset the system, and that's happening here at number 5, where all of the potassium and sodium are switching places to go back to what the original case was, with sodium outside and potassium in. Now we look at a propagation. This is where we get the idea of the wave, and you can see just in these 3 pictures how you can see the action potential is here in blue, and that's traveling from the very beginning of the axon to the middle of the axon and to the end of the axon like a wave. So first, we see that the normal situation here in red, again, when it's resting, we have positive outside, negative inside. When the action potential begins, sodium comes in, and that makes this part of the axon positive. It's not the whole axon all at once, though. It's just one little section of axon where the sodium gates open. But as soon as the sodium gates open here in this first section of the axon, then what's going to happen is that the next set of sodium gates are going to open up in response. So first the gates open on the left, then here in the middle. And that action is going to continue on down the line, so now you can see this third set of sodium gates has now opened. And that's going to happen all the way from one end of the axon, represented here on the left, to the other on the right, like a wave. Now what you can see here is kind of the second step of the action potential. After the sodium gates open and made it positively charged inside, now the potassium is getting pumped out. So as the sodium gates are opening further to the right, the potassium gates now are open here on the left. This, again, that second stage, pumping the potassium out and turning this part of the axon to having a negative charge. Okay, and we see that that continues on down the line. At the beginning of the axon, it's already back to its resting potential, negative inside, positive outside, where we have the action potential is still continuing on down these other sides. So that's going to happen, and it happens super quick. This is happening in a matter of a few milliseconds in terms of moving down the line of that axon, but still, it's not all the sodium gates open at once, not all the potassium gates open at once. It is happening kind of in stages, in steps, going from one end to the other like those dominoes. Now, once it gets down the length of the axon, well, as it's moving down there, one of the things that helps to make our axons a little bit more efficient in being able to send the signal quickly is the myelin sheath. And that's the purpose there of the sheath is to insulate the neuron, and that insulation helps to speed up the transmission of the action potentials. One disease that affects the myelin in particular is multiple sclerosis, and this demyelinates motor neurons. And so as a result, the neurons are less efficient and less effective in being able to send signals from the central nervous system out to your muscles, causing a deterioration of muscle function because of the fact that the nerve signals are not being sent as quickly as they should. The nodes of Ranvier are the spaces in between, and so when you have a myelinated nerve, what is happening is that the action potentials are going to be moving through kind of stepwise. So instead of those gates opening all along the whole length, what happens is that during every one of those little sheets, none of the gates are opening. And so the signal's being kind of sent skipping from node to node to node. So you can see how in the picture here you see the gates that will open and shut here in the space in between, and then where that myelin sheath itself is, there's not going to be any movement. And so as a result, we get a big increase here in speed from 150 meters per second in a myelinated nerve versus 5 meters per second when it's unmyelinated. Okay, finally, the nerve terminals at the end. This is where the impulse has to jump. So we don't have a physical connection between the edge of the axon terminal and the dendrite of the next neuron. They're not physically touching, and so we need some way to be able to have that signal be sent from one to the other. So here's where we define some new terms. We have presynaptic cells and postsynaptic cells. So the synapse is the space between. The presynaptic cell is the one that's sending the original signal. The postsynaptic cell is the one receiving the signal. So what's happening? Well, this is the end of the axon of the presynaptic cell. And as soon as that action potential gets here to the edge, it's going to open calcium channels. So again, here's another idea where, in this case, not salt is significant to our diet, but here we have the significance of milk or dairy in order to get a lot of calcium into our body. Calcium gates will open, calcium comes in, and that's going to trigger the release of neurotransmitters. Neurotransmitters are a chemical. They're going to exit the axon terminal by exocytosis. Once that chemical is released, you have receptors inside your postsynaptic cell that will receive the neurotransmitter. Once they receive the neurotransmitter, then they're going to absorb it and then generate a new action potential as a response. So neurotransmitter is just the chemical released from the presynaptic cell, received by the postsynaptic cell, and it acts as the mediator that allows the signal to be sent from the presynaptic to the postsynaptic cell. So there you have it. This is the interaction of neurons. So we had the dendrites receiving the signal, passing that on down the length of the axon by an action potential, and then you have the terminal of the axon releasing neurotransmitters, which are going to be received by the postsynaptic cells as the neurotransmitters bridge the gap, which is called the synapse.